Hello everyone. Thank you very much for giving me the opportunity to present our work in 3rd WBDS-LA conference. I am Abira from Indian Science and Technology Foundation and Moldoc Biotech Private Limited. The title of my work is Prediction of Endocrine Disruption Chemical Properties by In Silico Approach. So what is the endocrine system? The endocrine system is a message carrier complex composed of feedback loops of hormones, which are distributed by different internal glands of a living organism right into the circulatory system. It is responsible for production, storage, and secretion of hormones inside the body. The integral parts of the endocrine system include the pituitary gland, hypothalamus, pineal body, thyroid and parathyroid glands, thymus, adrenal gland, pancreas, ovaries, which are found in women, and lastly testes, which are found in men. Now, what are endocrine disruption chemicals? These are a class of non-natural chemical compounds which interrupt the normal functioning of the hormones inside the body. These chemicals can get inside our body through the contamination of food chain, inhalation of contaminated house dust, industrial solvents and their byproducts, plastic containers, pharmaceuticals, dietary components, personal care products, and even through the clothes that we wear. The effect of these chemicals include infertility, obesity, early puberty, insufficient growth, learning disorder, heart diseases, metabolic issues, less immunity, and so on. So what is the necessity of this study? Endocrine disruption chemicals are present in every single product that we use and eat in daily life, which disturbs the normal functioning of the endocrine system and may lead to hormonal disorder. Therefore, it is a matter of concern to predict the effect of such chemicals. Hence, in this study, we had aimed to determine the effect of such chemicals using in silico approach. Now, coming to the method of the work. First, we obtained the 3D structure of the estrogen alpha and estrogen beta proteins from RCSB databank. Then the non-natural chemical compounds were selected from literature. Then the selected compounds were docked against the proteins using autodock VINA. Then the post-dock complexes were analyzed in order to evaluate the affinity of compounds with the proteins. Now, coming to the results, in this study, estrogen alpha and estrogen beta receptor proteins were docked against a natural hormone estradiol and 3 non-natural chemical compounds, bisphenol A, bisphenol B, and hexastrol. Estradiol produced strong binding affinities with docking energy of -7.7 and -8.8 kilocalorie per mole against estrogen alpha and estrogen beta respectively. It was observed that the non-natural chemical compounds had comparable binding affinities with that of the natural hormone. Bisphenol A produced binding affinities with docking energy of -8.1 and -7.9 kilocalorie per mole against estrogen alpha and estrogen beta respectively. Bisphenol B produced binding affinities with docking energy of -8.2 kilocalorie per mole against both estrogen alpha and estrogen beta respectively. And hexastrol produced binding affinities with docking energy of -6.2 and -8.5 kilocalorie per mole against estrogen alpha and estrogen beta respectively. Therefore, our study confirms that the non-natural chemical compounds compete with the natural hormone to interact with the estrogen alpha and estrogen beta receptor proteins and interfere with the normal functioning of the hormones. Now, in conclusions, we can say endocrine disruption chemicals are present in every single object that we use in daily life, which may lead to hormonal disorder. Therefore, it is a matter of concern to predict the effect of such chemicals. In this study, estrogen alpha and estrogen beta receptor proteins were docked against a natural hormone estradiol and 3 non-natural chemical compounds, bisphenol A, bisphenol B, and hexastrol. Estradiol produced strong binding affinities for both estrogen alpha and estrogen beta proteins. Non-natural chemical compounds were observed to have comparable binding affinities with the natural hormone for both estrogen alpha and estrogen beta proteins. Therefore, our study confirms that the non-natural chemical compounds compete with the natural hormone to interact with the estrogen alpha and estrogen beta receptor proteins and thereby interfere with the normal functioning of the hormones. Therefore, we can say that the in silico approach can be valuable in sketching the in vivo assessments for predicting the participation of the non-natural chemical compounds in endocrine disruption. Now, I would like to thank Inside Biosolutions France for providing all the support. Indian Science and Technology Foundation and Moldak Biotech Private Limited for letting me work on this project, and also this conference for letting me present my work. Thank you so much.